Hi, and welcome to the Anti-Waffle podcast with Em and Pixie.
Woohoo!
And together we will try to talk about research without any gobbledygook. Each episode we'll be talking to a researcher who will be discussing the work they're doing, looking at why this research is important, and asking how did they get into this field. We hope this will be informative and interesting and bring a bit of the academic world into the real world. Today we have Professor John Ward, Professor of Synthetic Biology for Bioprocessing at UCL. Hi John.
Hello.
Hello. Thank you very much for being here and talking to us today.
Not at all.
Thank you. Would you like to start by telling us about your current research?
Oh, sure, yes. Well, currently I'm working on a long-term project using enzymes, and those are the proteins that do all the individual bits of chemical reactions in cells. And we're using those to make very specific changes in small molecules as a way of doing with biology what organic chemists do with chemistry. And so that's been going for about 10 or 15 years now on various different types of enzymes for this kind of area we call biocatalysis.
Biocatalysis?
Yes.
So what's biocatalysis?
Well, it's catalysis is a chemical name meaning that you're using a certain chemical, usually a chemical, to effect a change in a molecule. And a catalyst is a chemical or an entity, and it can be something like silica, which is sort of like a rock, which is unchanged in that reaction. So it reverts back to its first state, but the chemicals it's changing go from being one chemical to another, and that's what organic chemistry uses all the time. So catalysts are used in industry, they're used really extensively to effect a very specific change from one molecule to another. And it could be a very minor change, like putting just one atom into a small molecule. And organic chemistry has done that for ages, but it uses a lot of things like organic solvents and heavy metals, which in industry you have to get rid of afterwards. And so nowadays there's a big move to making that more sustainable and greener. And one way of doing that is to use enzymes, so biological catalysts, to do that.
Ah, interesting. Because when I looked it up, the synthetic biology, now it made it sound like that it was that you were looking at processes and then looking for ways to change them. But then it was, so to me it sounded like you were just messing around with the human body and trying to get it to react in different ways. But then I'm like that. I'm very much what are you doing to a scientist? You know?
Well, synthetic biology is a sort of an overall term that has many different parts under that umbrella.
Oh, right.
And the part I've just described with using these enzymes, these biological catalysts, is one of those types of what we might call synthetic biology nowadays. The kind of synthetic biology that you might have looked up is what people often look at as the more controversial parts of synthetic biology, where nowadays we can take DNA and we can actually synthesize that. Companies will synthesize long strands of DNA that in the lab we can then stitch together into longer strands until we can make whole genes and even much bigger DNA elements, almost whole, well, we can make whole chromosomes now.
Right.
But that takes many, many people and a number of years, so it's not something that you could do just on the bench upstairs.
Right. But they might have started it 30 years ago.
Well, the synthetic biology revolution really started with some of these new capabilities of being able to synthesize DNA. So if you go back even just 10 years, there wasn't the availability at the right price of DNA. And there's been huge technological revolutions in many fields. One is DNA sequencing. So we can sequence genomes now very, very fast and very cheaply compared to the original.
Sequencing genomes, is that like just reading them, saying
exactly
what they do?
Yeah. So it's using, and nowadays it uses very complex and quite miniature machines. You put a bit of DNA in and it will literally decode the A, T, C, and G code of that DNA. And that has gone through 3, and possibly now even a 4th, major revolution in the technology. So when I was a PhD student and then a postdoc way back in the 70s and 80s, last century, we did that by hand. And in fact, when I started as a PhD student, the first papers describing how to do DNA sequencing were only just being published. So in my research life, it's gone from being a technique where several people got their Nobel Prizes for that, all the way through to now we send our DNA off, and my research group are doing this, multiple samples every day, send it off. A company will sequence for about a tenner a certain amount of DNA. When we want whole genome sequence, it's a lot more money. And then we have to spend quite a long time doing what we now call bioinformatics, where we sit at a computer and try and do quite a lot of analysis on that. But we're getting little bits of DNA to check our day-to-day work in the lab. We're getting those sequenced every day. And so that's revolutionized what we do. So what used to take me and several other people virtually all of a PhD to get a few kilobases, that's a few thousand bases of DNA sequenced, is now done by return of post almost.
Oh, wow.
And that it doesn't come in the post. We send the DNA off in the post because it's a physical bit of DNA. And electronically in our inbox we'll get a DNA sequence about 2 to 3 days later. And we're sending multiple samples of those every day just to check our day-to-day work. So that's become a completely normal work-a-day thing, getting DNA sequenced at a very cheap price. And then coming along, sort of not quite in the same sort of speed of technology, the DNA synthesis, the actual building of DNA to make completely synthetic long strands of DNA, that again has undergone a few changes, but it's still a little bit more difficult to do than the DNA sequencing. But the cost of that has come down. We'd like the cost to come down even more, because then we could build longer things cheaper. But it's sort of a price now where in many projects I'll say to the researchers, let's go and have that gene made, because it's actually quicker than you finding the genome that gene is coded in, and then we have to do a technique called polymerase chain reaction to amplify it. And in some projects, time is better served by that person doing some other experiments at the bench, ordering the DNA, and so while they're doing their other experiments, that DNA is being made for them. 3 or 4 weeks later, it takes a little bit longer to make DNA than to sequence it, well, quite a bit longer. And then they can receive in the post, and again, it is a physical post. You get a little tube with some what we call lyophilized powder, that's freeze-dried. So a tiny little smear in the bottom of a tube, which is then the DNA that we've ordered. But we're getting that made, again, every week or so we'll order some synthetic genes as a way of either speeding up a project or accessing DNA constructs that we couldn't actually make. So we can then make completely synthetic constructs. And in doing that, we can change some of the parameters of that, if you like. We can then put different amino acids in, or the coding sequence for different amino acids into the gene. We can put different signals at the beginning and the end so that it'll become what we call expressed. It'll actually make that protein that it's coding for in possibly different cells than it came from. And so it's quite possible now to build really long strands of DNA with multiple genes. We would probably order that in several segments because it's actually cheaper that way, quicker, and it's easy for the companies to make it that way. And then we would stitch it together in the lab, which we've done with a few of our projects.
So there's actual companies out there that just concentrate on making up these?
Yes, many now, yes. Yes. There are 3 that we routinely use, and I've got an account which I top up from different research grants, with one called Eurofins. It's now, in the time I've been using it, it's taken over or merged with 3 other companies. It's the company that actually had a major cyber attack several months ago, and it carries out all the forensic science sequencing for the UK.
Oh.
But it also has about 3 or 4 other major jobs under its sort of umbrella. So it's not just making DNA and sequencing DNA. It does quite a lot of other forensic science and other genetics services as well. So some of these companies have grown to become quite big, sort of multifaceted companies.
Right.
And there are several others in Europe, and the other big ones are in the United States.
So for your research, is there an example you can give us of what it's used for, or something so that we can sort of understand how it fits in to our world?
Okay, so the area that I talked about to begin with, biocatalysis, and we're using these synthetic genes in that project anyway. We use synthetic DNA and sequencing DNA in all of our projects, so that's become just a tool.
Right.
A series of tools that we use.
For
testing and building things, we can build new things with the DNA. So we can look at, so for example, one of the enzymes, and enzymes are proteins.
Right.
So proteins are and enzymes are the workhorse of all cells.
Yeah.
So the DNA is the information storage, and that has to be made first to be able to then what we call express the protein. And so the information in the DNA is decoded inside the cell, and that could be a bacterial cell that we use in the lab, or human cells or plant cells. So the DNA is the information. It's then read, and we use these terms loosely in the lab, in our jargon. That's read into an intermediate molecule called mRNA, which we never really touch. It's what we call labile. It's made and used very quickly within the cell. We never have to touch it. The key thing that we want is that mRNA is then translated in the cell into a protein. Right. That's its job. So DNA goes to this intermediate messenger RNA and then is made into many copies of an enzyme or a protein or whatever the gene encodes. And it's those proteins that are both the structural components of all cells. So if we look at our cells, you can see skin. Yeah. And those are major sort of structural proteins all cross-linked to give us a barrier to the outside. And then all of the enzyme reactions, all the metabolism that's carried out in all cells, from microorganisms through to us, to plants, to blue whales, are carried out by these enzymes. And enzymes are proteins. They're proteins that have a very special, usually a very specific chemical job to do. So all of the metabolism, from eating food, breaking it down, making energy for us, or the ATP that's made, and then making and building more of us, is all done by these enzymes. In the human genome, there's people don't know exactly the number, probably about 20,000 different enzymes that are potentially able to be made by the human genome. Maybe a few more, it depends who you ask. And that's where it gets very complex with how you use bioinformatics, i.e. computer techniques, to actually work out how many genes there are in the human genome. That's quite a difficult job because the human genome is very complex. In E. coli, the bacterium that we use, the day-to-day lab bacterium, we know exactly how many genes there are. And different strains of E. coli that we use in the lab, usually the ones we use have about 4,500 genes. So that's 4,500 different proteins that can be made.
Yeah.
They're not made all the time because even an E. coli cell, a small bacterium, is very sensible. I don't think we'd use the word clever because that means it's sort of some kind of intelligence, but it's very sensible. It will have a lot of its genes what we'd call turned off, i.e. they're not being expressed into protein because it doesn't need all those genes are on all the time. There's a whole series of genes called the housekeeping genes, which are the genes for replicating the chromosome, making the protein, making the apparatus that makes the proteins, making the cell wall, constituents, things like that. And those are on usually all the time because the cell needs to grow and be itself.
Yeah.
And then it'll have a lot of enzymes that are only going to be expressed when a particular foodstuff is in the environment, or if it's got a particular job to do. So that job might be to transfer DNA to another cell. So then it'll start to make a whole series of proteins, enzymes that will process DNA to go to another cell. Okay, I've got off a bit of the topic. You asked about a particular project that we're doing. Okay, so going back to these enzymes, one of the groups of enzymes that we've been using extensively over the last few years is a group called transaminases. So their job inside, and they occur in most cells, is to move a nitrogen, usually as a little, what we call an amine, so that's nitrogen with two hydrogens attached, from one molecule to another. And there are many types of those within most cells because nitrogen is a very important key element for all life. So carbon, nitrogen, oxygen, and hydrogen are the main sort of elements of life, and then you have very minor things. And we need nitrogen all the time. So we eat that in the form of proteins. Proteins contain amino acids, and most organisms have ways of balancing their amino acid range that they've got inside the cell. So if you're eating just one kind of food and it's rich in certain amino acids but lacking in others, many cells have ways of swapping that NH2 group, the nitrogen, from one amino acid to another. It's a little bit more complex than that, but that's essentially what's happening. So that it'll balance its range of amino acids inside the cell, and then it can use those to make more proteins or to metabolize and make other nitrogen-containing compounds. So cells have lots of transaminases. So transaminases are very useful because they will put this nitrogen group into a small molecule. Sometimes they'll put it into a non-natural molecule. Now, we use the enzymes outside of a cell, so we will prepare genes that will make large quantities of a particular transaminase enzyme. We'll grow that in E. coli in the lab, big liter of broth that's growing. We'll express the genes. We'll express the transaminase that we grow in that E. coli, in that liter of broth, purify the enzyme, and then we've got essentially a single enzyme in solution, free of all the others, and we can use that to do a very particular bit of organic chemistry. And that's where we can use an enzyme as a replacement for doing it by a chemical step. And often with putting an amine into a molecule, it's very important in, say, making pharmaceuticals, because the presence of nitrogen or an amine in a compound often gives it biological activity. Right. So most of the drugs, small molecule drugs that are made, have at least one nitrogen in. And that is because most of the hormones have nitrogen, especially if they're protein or peptide hormones in our bodies. So the receptors on cells are all geared up to bind nitrogen plus other parts of a molecule. So of course, drug companies will make pharmaceuticals that either mimic those or lock themselves into a receptor and stop the activity of a cell. So you can have things that will turn off a cell. Okay. So in cancer research, we want to do that, or activate a cell to do something. And very often, those small molecules are binding and mimicking the binding of a naturally occurring molecule. So for an example of that is morphine, a very powerful pain-killing compound, mimics or binds to the receptors of a naturally occurring little peptide that our brains produce, a thing called met-enkephalin, and there's another one called leu-enkephalin. And they're quite short little peptides. They bind to receptors in the brain, and they're part of the sort of pain reception and communication pathways in the brain and the rest of the body. Plants have evolved years, probably billions of years ago, ways of making compounds that slot beautifully and specifically into the same receptors and bind very tightly. Probably in the war against plants and insects, the compounds were made as a way of preventing some insect eating them.
Right.
And what we see in biology is that there is a huge amount of shared biology. So the receptors that are in those insects, although they've become a little bit more complex and there are many different subcategories of them when you get to humans, those similar receptors are there, and they're still going to bind the enkephalins or the morphine, and that's how plants actually make compounds that, strangely enough, bind very specifically to our receptors. And many drugs are either based on these naturally occurring compounds, or they're minor modifications of those. Or indeed, pharmacologists and people in the pharmaceutical industry have used those plant-based molecules as the lead compound to then make synthetic compounds that sometimes now don't look much like the original plant-based, but they know, yes, this compound binds tightly to a human receptor, so we'll also try and find other ones that bind and then make new ones of those.
So did you say that you're working on designing these enzymes, the catalysts, the
transaminases?
Transaminases. So you're creating transaminases that can replace or do what the ones that are in nature anyway, or that are there anyway, but do it better, or you're taking that design and using it, applying it somewhere else.
Yes, so we will use we start often with an existing transaminase that we'll clone and express in E. coli, many different variants of those from many different microorganisms. And what we do is put it through what we call a screening process, usually on a compound that is completely non-natural, but it'll tell us whether that enzyme, out of a panel of enzymes, which one is able to then put an amine group into quite a complex molecule. And then using that enzyme, we often then start to change that, either mutate the gene in E. coli so that we can then rescreen and find ones that then will be either much tougher. So industry and we in the lab would like ones that are much more stable. And stability is very necessary if you're going to use something in industry. You don't want something to be only able to be used for like 10 minutes and then somebody has to go away and make some more.
Right.
So we want things that are thermally stable, because often processes might happen at either a higher temperature, or even if they happen at room temperature, we'd like an enzyme to still be active 24 hours later and still be able to do its activity. In industry and in our work as well, we often want them to be stable in the presence of organic solvents. Many drug molecules are not very water soluble at all, so you can't really use all enzymes work in water basically, or a buffer.
Right.
So we would like our enzymes to be able to tolerate, even though they have to work in water, tolerate organic solvents, often like ethanol or methanol. And those are used in industry, and they're what we call green organic solvents because they are biodegradable. They're easy to dispose of. But industry needs those to dissolve the compounds that it's working on. And if we have enzymes that can also tolerate those organic solvents, then we've got the best possible combination of specificity, because enzymes give great specificity, stability, tolerance of the solvents, and then we can start to use these in what we do in the lab now is build multi-enzyme cascades. So that's a series of enzymes where We can start with a simple compound, and the first enzyme will transform it into its product, and that is now the substrate or the starting compound for the next enzyme in the pathway, and so on. But these are outside of a cell, so we can put them together in just literally in a little plastic test tube, multiple enzymes carrying out what we now call cascade reactions, and we can put in a starting simple compound, and through a series of steps, and at the moment we've got 5 enzyme cascades going routinely, and out at the end comes quite a complex molecule with new rings being made, nitrogen and other elements being put in very specific places. What we're doing is not trying to make a drug. We're not aiming towards a single drug. That's for proper pharmaceutical companies to do. We're showing what can be done. We're making almost toolboxes of these enzymes to be able to say, look what's now possible, because this has not really been done before, so we're sort of showing this is what biology can do. And that's where the synthetic biology comes in. Because that's really saying, right, what can we use all these biological reagents, because the enzymes are reagents, what can we actually use them for? Can we build this complex compound? So by sort of pushing ourselves to really make quite complex molecules, or make the enzymes work in very unusual molecules, we can show what's capable, and then it's up to others to then use those ideas and take them further.
Is there a particular application that you've already, where we said, oh yes, no, this is what we created, and the industry have actually sort of like taken it up already? And so you said, okay, that was a success. Now we move on to the next thing. Have you got one that's...
Well, one of our transaminases that we discovered, we tested a number of years ago, was handed with all the appropriate sort of agreements to a collaborating company called Almac, and they are now currently using that in one of their processes. Now, of course, they don't always tell us exactly what molecules they're making, because they will be using the transaminase on another company's compound, because Almac are a company that make compounds for other drug companies. But we know that our enzyme is being used by that company to make new molecules.
Right. And so do you see, as the end picture that what you're doing will make with drugs, make the actual making of drugs or the development of drugs easier?
Yes.
And more.
Yes.
And will it make the drugs themselves better, or?
In one way, it will make the drugs better. In all of the legislation for pharmaceutical companies has moved towards making what we call single enantiomer drugs. And that means that when in organic chemistry, when you're making certain kinds of steps, the organic chemistry can put a new atom in one of 2 orientations. And in fact, it's sometimes quite difficult in organic chemistry to make it only in one orientation. So now there is a move to having only single isomer or single enantiomer drugs, which means that the FDA, that's the drug licensing authority in the States, and the one in Europe and the UK, will now require manufacturers of drugs to make only single types of these, what we call single enantiomer drugs. Now that is very difficult for chemistry to actually build those molecules so that every single point where it could be in an atom could be in one of 2 orientations. Making it in only one orientation is very costly often. It often needs many steps just to get that one atom in the one orientation. Now that's where enzymes can really help, because they are uniquely able to make single enantiomer constructions or steps,
right,
because that's what they do in all metabolic pathways. So all the amino acids that are made and used are only of one enantiomer type. Right. We call them L amino acids. All the glucose that we eat from plants is what we call D-glucose. And it could come in D and L forms, glucose, if you made it chemically, which would be very difficult anyway. But plants only make one type of that. And our bodies and the enzymes that take it up and then metabolize it only accept one type. So biology is already uniquely able to both make and use these single enantiomer compounds. That's how the whole biology is set up like that. Which means the enzymes are already making and using these compounds as single enantiomers. So that's where enzymes are uniquely able to then substitute often several chemical steps for a single enzymic step. And indeed, that example I gave of this company, Almac, their process had, at the point that they were going to use the transaminase, it was replacing roughly a 7 or 8 step part of their synthesis pathway just to get this amine group into a molecule in the right orientation. And because often you have to block certain other parts of a compound, then effect your change, and then unblock bits that would have been attacked by the because chemistry often uses rather powerful reagents that will attack other parts of a molecule. So in chemistry, there's a lot of protection and deprotection steps. And that's called atom inefficiency, because you have to put a lot of things onto a molecule, which you then take off, and they're not part of the final molecule. They're there just to do protection, then you can effect one change in one part of the molecule, and then you deprotect and actually throw away all those bits. Whereas an enzyme doesn't need to do that. They will only bind to one part of the molecule, effect their change, and they'll effect it in one orientation, and then the molecule is no longer a substrate for that enzyme. So enzymes are fantastic when it comes to thinking about how to build new molecules. And in fact, in plant and bacterial metabolism, there are huge numbers of very complex molecules made, and they're all made by enzymes. And some of those compounds cannot even be made by organic chemistry. They're still just too complex to make. Right. Some of the current things we're doing, along with collaborators in chemistry, but also across several other departments at UCL, we recently got a project from the EPSRC, that's one of the UK's research funding organisations, to look into the problems of plastic and plastic waste. So our part of the project in my labs and in the chemistry labs are to try and find both bacteria and enzymes that can start to degrade and chop up, very specifically, certain kinds of plastics. Right. So
That's
one
good,
of the
isn't it? Yeah.
Yes. So one of the plastics we've started with is called PET or PET, and in fact that's the clear plastic that all water bottles are made of. And in a way that is a slightly easier problem than some other plastics, because there are already enzymes and bacteria that were isolated probably about 5 years ago that are known to be able to break down this PET, this PET polymer. But they can't break it down very quickly. And even if you had a vat full of the bacteria and you put a plastic bottle in, it would take a long time to degrade, because the polymer is crystalline at room temperature. So it's like something coming to a hard, rocky crystal and trying to eat it up. I mean, it's not very easy to do at all. So what we're looking at is to try and make mutations in the genes for those enzymes and make them better to work under different conditions, higher temperature, for example. And we just started, and it was announced today, we've got a sort of parallel part of the project is a sort of composting, a citizen science composting project, where you can log on, register, and at home you can then, if you do any composting at home, in a garden, or even indoors, what we're looking at is to see if biodegradable plastics, which are now starting to come into the market in foods and cafes, things like that. They're made of different polymers that are theoretically biodegradable. And what we want to do in the Citizen Science Project is to see how fast they degrade and under different kinds of real home composting kinds of situations. So whether it's an outdoor compost bin or an indoor one, those will be at different temperatures. There'll be different materials going into them, food waste and other things. What we want people to do is to log on and take part in putting in certain kinds of items and logging when they put them in, what they were. It could be a biodegradable plastic cup that you now start to get, or you can get knives and forks that feel as though they're made of plastic, but they're made of these PLA, polylactide plastics, and there's other kinds of biodegradable plastics. And we're going to see if 6 months later, if people empty their compost bin, is there still a plastic knife in there? Is it recognisable? Has it completely disappeared? Because there's a little bit of confusion now about the words compostable, biodegradable, and whether they really mean at home in a sort of low temperature compost heap, or in what we now have in many places, these municipal composting facilities where household waste is collected and it's put into very big piles, meters high, hundreds of meters long. Often they're in types of facilities where air is forced through those to raise the temperature. So you get metabolic activity of bacteria degrading those, and the temperature rises to sometimes 60, 65 degrees. Now, under those conditions, a lot of things will compost, biodegrade, that wouldn't happen at home if we stuck it in our home compost. What we want to do is to see what really would happen in a whole variety of different real composting situations.
Yes.
Okay.
And compare that with this sort of municipal composting system.
Right.
Wow. And so the project's just started now?
It was announced today. We've been planning it, and me, Helen Hale, several others have been involved in working out a questionnaire. Also, the kinds of things that we might ask them to record, maybe a picture of their compost heap, and then how we might then interact with them.
To record it audibly, and it's like, I'm sure it won't make any noise. Oh, this is it degrading.
Actually, my one at home, because I've got a couple in the garden, if you open the lid, there's a lot of low-key noises because it's full of worms and little insects. If you put your ear to it, you can hear all this slithering and sort of slimy. That's what you really want. You want a lot of life in your compost and know it's working well.
Yeah.
And of course, the things you can't see in there are all the bacteria and fungi that are doing often a lot of the initial breaking down. And those are the things I'm interested in, the bacteria especially, because I will then have a compost that we've set up one in Gordon Square, just outside UCL, and we're putting into it shredded PET, so shredded bottles of PET and other containers from collecting from the department and my own house. And over the summer, we're collecting those. We're now shredding them in a departmental paper shredder that they've let me use. We've also
it's like the plastic bottles you just push into.
Well, you have to cut them up a bit first, otherwise you can't
fit it through the hole.
No, it soon jams up if you try and do that. So that's taken us quite a while. And what we're also doing, we've set up some enrichments in my incubators upstairs. So I've got a 50 degree and a 65 degree centigrade incubator upstairs, and we make what we call enrichments. Is basically we've got a large glass flask, for example. We'll put in some certain kinds of composts that already we know have some thermophilic organisms in, chop up some plastic bottles, literally plastic bottle chopped up, a little bit of water plus some mineral salts and some other things as well. And then let that stew. And some of those have been going all the way through this summer in an incubator upstairs. We are testing that every now and then, take a tiny bit out, and we'll do our agar plating studies to see how many organisms are growing in there, what kind they are they actually degrading PET, and that's quite a difficult thing to determine. And some of those enrichments, we're then taking small amounts of those, we'll expand them and grow them up a bit in some nutrient broth, 50 degrees, 60 degrees in the lab, and then we pour it on top of this compost bin. Well, not on top of the bin. We pour it on top of the compost inside the bin and leave that to go. And so that's only just being set up in Gordon Square. And it's a certain kind of, it's called a hot bin. It's got very thick insulating walls. So it's designed to contain the metabolic heat that bacteria and fungi will start to generate in any compost or soil. If you can retain all that heat, then naturally it'll get up to 50 or 60 degrees centigrade. And if it stays there at that temperature for a number of weeks, we hope, then the metabolic activity and the enzymes will then be able to work faster and degrade, we hope, the PET. What I'm looking for there is new novel bacteria that we can look at and isolate. Also to see if we deliberately put in bacteria that already have come from enrichment. Does that mean the PET bottle fragments are broken down faster?
Right.
And we'll probably set up at least one other of these hot bins with different material, different plastics that we're interested in getting degradation of.
Yeah. That's very exciting.
Yes. There's the one in Gordon Square. Can people see it? Oh, is it like
Yes,
open to the public?
yes.
Can they put things in it?
No, I'd rather they didn't, because we are putting very special things in there.
Yeah.
And in fact, some of the feeding that we're doing, we're autoclaving, we're sterilising food waste first.
Right.
So I'm collecting that from the department, but I'm sterilising that in our big autoclaves, which are huge big pressure chambers, like pressure cookers, so that what I'm feeding the bugs that are in the compost is lots of nice foodstuffs, orange skins and bits of somebody's takeaway meal, but they have no bacteria in that we don't deliberately put in ourselves.
Yeah,
So you
okay.
can control. Right.
So is there a sign up that tells people what it is?
Yes, it says UCL Plastic Innovation Hub. For any information, contact j.ward@ucl.ac.uk. You can take a picture of it if you want.
Yeah, let's go around and have a look. Yeah. And when's the project running until? How long are you getting the
Well,
data?
the EPSRC funding will only go till June 2020, but I've been doing things like that for many years, and that will continue beyond there. So that won't just stop when that grant stops. And we're applying for some more funding at the moment as we speak.
So can people get involved and then?
Well, the Citizen Science Project is for people to do at home.
Right.
So they will register and they'll say, yes, I will log on to this and I will sign up, and here's my compost bin and I will put these things into it, and then 6 months, whatever length of time, they will then look at their compost, because you have to then take it out and do things. And they will log or record what they then see. So they'll hopefully take pictures of what they put in, literally a biodegradable plastic cup, spoon, fork, knife, and then 6 months, a year later, show us they will record what they've found in their compost and tell us a bit about the conditions. If it was outside, did your compost get warm? Did it have lots of worms in, for example, so it shows it was really active? Or was it inside and was it just food waste, or did it have garden waste in, that kind of thing? And then we can start to make some kind of assessment of, under these conditions, nothing degraded, but under those conditions, maybe all things degraded.
Oh, that's quite good. I could do it with my kids.
Yes, please do, yes. Because we're wanting a good spread both of different conditions. I think the more different conditions we have, the more different kinds of composting, the better.
Yeah. And are you asking people to report back in 6 months, or is there a certain time?
No, there isn't a specific time.
Right. Okay.
But I think we've suggested certain kinds of things, because some people will have their own procedure, and not like me at home. I've got a big compost bin in the garden, and it takes all our food waste, but also stuff that we trim plants and shrubs in the garden. We don't put grass in there. That usually swamps a compost bin. But I'll wait till that's filled up to the top, and then I will not put anything more in. But that takes about a year, and then I leave it for another year, because I've got 2 compost bins, so we can rotate them. It'll be about another year before I empty it. And during that second year, when you don't put anything new in, and you just leave it, a huge amount of things happen. And by the time you then open it, it's all completely black, friable material, just like, it's not quite like soil because there's no mineral content in, but it's very rich, and you can add it to flower pots and put it on the soil, things like that.
Wow.
Oh, I'll look into that.
So in that case, I wouldn't be able to look into it for 2 years.
No.
But other people will have maybe a shorter cycle time, especially if you're doing things indoors or have a much smaller bin outdoors. It might be 6 months.
Okay. But it's so topical, isn't it, at the moment? The whole, we found to find something that was breaking down the plastic.
Yes, I think everybody's really aware of the kinds of waste that we throw away, and when that gets into the wrong place, like landfills, spills out, goes into the oceans. It's a very visible problem. A lot of the plastic, like PET, is very well recycled, especially in the UK, because it actually has some value. And so we've, in parts of this plastic waste hub grant, we've been to visit various recycling places. So one in South London, where they take all the waste from that local authority and do the initial sorting. So the factory literally has hundreds of conveyor belts with all kinds of teeth and sprockets and things, and waste goes in, and this is all the recycling waste. It's all the sort of dry paper, plastic, cardboard, metal, and by the time it comes out at the end, through all these complicated interconnecting conveyor belts doing all kinds of things, it's separated into all the different things. So you've got paper, cardboard, and all the different plastics. So the plastics are then squished into great big cubes almost, and then they go to a specialist recycling factory somewhere. And that's value, because that material can be made into new plastic bottles. Nice. So most of what we call waste, if you can recycle it, it has value. So it's then not waste anymore. Mm-hmm. It's a resource. Mm. Now we're quite used to, in my childhood, quite used to recycling glass bottles. They've almost disappeared from shops now.
Yeah.
But things that we're familiar with now, paper, cardboard, they're eminently recyclable, and they have value, which is why there's a paper collection, there always has been. And nowadays, with everybody getting orders online, the amount of cardboard that exists is enormous. But there's a lot of money in taking that product, recycling it back into more cardboard, and it can be easily recycled back. So anything where there is value in it, then it can be recycled into either the same product or other products. So to maintain the clarity of PET bottles, you always need quite clear plastic to start with, and it'll often need a little bit of new, what we call virgin PET, to put in there. And if you have any coloured material, that gets then taken away and made into fibres for garments, things like that. Okay. So, and you know how they say, oh, this jacket was made from 18 recycled water bottles. Yeah. Well, that's happening all the time now, because that's actually worth quite a lot of money to somebody, because then the jacket's worth, I don't know, £50, and it's come from a lot of recycled bottles. Yeah. And you don't have to then buy, or the company doesn't have to buy brand new polymer from a big petrochemical refinery. So there's sorry.
So if it's got value, why are you working on PET first? Why are you working on PET first? Why didn't you start like with carrier bags? Because they're not, are they recyclable? Like the ones you get in the shop?
Well, there you reveal what we do is work on the things that we can work on easily first. Mm-hmm. So I said a little while ago, a few years ago, scientists in Japan, I think were the first ones, discovered bacteria and some enzymes that bacteria secreted that could degrade PET. And that's why we're working on that, because there's something immediately able to be used and worked on. The other plastics that you mentioned, carrier bags are made of usually polyethylene. There is no known enzyme system yet for degrading polyethylene. And because it's very flimsy, it's not got a great deal of weight, so you actually have to gather very large quantities of plastic carrier bags to be able to then gather together and make into something else. But there is value in that. But biologically, it's much more difficult to degrade. And the same with polypropylene, and then other polymers like expanded polystyrene. There's a lot of that around, but again, there's no enzyme system for that. Now, part of our research is to initiate work on those kinds of polymers.
Right.
So that's not yet at a stage when we can say, oh yes, we've got lots of systems to do this. And the PET systems are much more far advanced. So there we could start to make some inroads into the waste stream of PET. Some of that PET is recyclable. Once it gets down the sort of value chain, say it gets made into garments, textiles, you can't do much with it after that, because if you then chop it up and do things with it, it's then got a lot of dyes in, it's got a lot of other products in, so it then actually eventually gets into something that will eventually go into possibly landfill. So if we have enzymes that can then start to do something on that particular part of the waste stream. So what we're not doing is develop enzymes that are going to chew up plastic bottles, because in the UK at least, I think greater than 90% of all the PET bottles are recycled, because they're actually got quite a high value in that kind of industry. But when you get that PET polymer that's been then made into textiles, and it's been made into something else, it's no longer able to be made into bottles, because it's now got dyes in, it's got some other things in. We would like to be able to say, right, now at that point we could have an enzyme or a microbial system that could do the following to it, break it down, and maybe then depolymerize it into its constituent monomers, and those will have value. They won't have value, well, they might have value to make back into bottles, but they could have value to put into the as the starting point of a chemical factory to build new molecules, not just plastics. Those other plastics were right at the beginning of being of trying to work out how to use biology, enzymes, bacteria to depolymerize things like polyethylene or polypropylene. That is really quite a big ask, but we're just starting on that.
How did you get into your research? How did you get to this point? Where did you start out from?
Story's quite a sort of long and convoluted one. I mean, I was a biochemistry undergraduate in Bristol, and then I moved department, stayed at Bristol to do a microbiology PhD. And at the time we were looking at antibiotic resistance genes and how they move between bacteria. So I learned a lot of the molecular biology techniques there that sometimes I still use, looking at DNA on gels, things like that, and being able to start the manipulation of DNA. So that got me interested in being able to use biology to sort of build new bits of DNA. I moved to Manchester, and I started working on a bacteria that could degrade toluene, which is an organic chemical. And it does that because it carries a very large circular DNA molecule called a plasmid. It's not the genome or the chromosome of the cell. It's a sort of extra chromosomal element called a plasmid. But that carries all the genes for toluene degradation. So I was very interested in the enzymic sequence of steps for degrading toluene, which is an organic chemical which is used as a solvent in industry. And bacteria can degrade that. They're quite happy to live on that as a sole carbon source. And the enzymic steps there are very interesting. There's lots of interest in certain steps because they introduce oxygen into the molecule very specifically. So I got then an interest in biocatalysis, using an individual enzyme that might have come from a long sequence to do something else, but then take one enzyme out and see in the lab how many other different subtle changes to the compounds it could deal with. And then coming to UCL many years ago, I started some projects that were on other kinds of biocatalysis. So my interest was in manipulating the genes, the enzymes, looking at how these enzymes can affect really interesting bits of chemistry. And then more recently, teaming up with colleagues in chemistry who can do the analytical part that I can't do, and also make very specific compounds I can test the enzymes on. And that's a very fruitful collaboration. Then expanding my interest in lots of different enzyme areas. So the transaminases started a while ago because there's so many of those and we can do lots of different things with them. And then a few years ago we started this project on, we made a synthetic enzyme that currently exists only in plants. So we made a synthetic version which will work in bacteria, and that will start the process of making alkaloids, which are a family of very potent plant drug type compounds. And then using all our other enzymes, the transaminases and several other kinds of enzymes, we can make these, what I described as cascades, where we can make really quite complex molecules like these novel alkaloids using enzymes. So it was just my interest in microbes, what they can do, how they do it with these enzymes, and then understanding how you can then manipulate the enzymes, put different ones together in a test tube and actually make these complex cascades.
How can you sum up why your research is important to the public, if you could do it in a short sentence or paragraph?
Well, I think it would be to not only show how you can build new things using enzymes, which are much more sustainable, greener than using chemistry. That's really what I would say is being able to build new things but using greener and more sustainable enzymic techniques.
Brilliant. Anything else you want to ask?
So are you like the natural enemy to chemists?
No, I collaborate extensively with chemist Helen Hales in UCL chemistry department. We need chemists because we analyse all the materials that we make, both the compounds that we're starting with and then the new ones that we make. We have to analyse those with really quite in-depth chemistry techniques. Mm-hmm. Very often we have to synthesise chemically certain compounds that we're going to use in our testing of the enzymes. Not all of the compounds are commercially available to buy, so we'll have to make those. And then the understanding by chemists of what the enzymes might be doing, it's a two-way process, so it's constant backwards and forwards. So no, I think that would be a wrong way of looking at it, that we're trying to supplant those. What we're trying to do is also show to others, both academia and in industry, how, oh look, you could use an enzyme for that step and you could then do away with 6 steps of chemistry in industry, but use an enzyme for that step. That causes a few more or slightly different problems. You'd have to make the enzyme, so actually you'd have to have companies that could make enzymes in large scale. So that could either be the pharmaceutical company or it could be a second company that just supplies enzymes. So it actually is the beginning of sort of building new infrastructure, companies that make enzymes. But I see the need for, we all want new drugs. We need, if you look around here, we've got dyes in all these materials. All those come from what you'd call a chemical company, but many of those could now be, or in the future, might now be made biologically with enzymic steps. So I'm looking at, and this is a long-term thing, it's not going to be changed instantly, perhaps sort of the beginning of this sort of biological revolution, which this century will actually be, to start to make many things, our textiles, our dyes, our drugs, with many more, in fact, perhaps exclusively biological steps.
Yeah.
Thank you, John. Thank you very much for your time. Thank you to everybody for listening. We'll put links up on our website, which is antiwafflepodcast.wordpress.com, and our Twitter account, which is @antiwafflepod. Please do get in touch, as we'd love to hear from you. Bye.
Bye.
Cheerio. Bye-bye.